Assalamu alaikum friends. Hope so you all will be fine. Today's the topic of our discussion is cholesterol biosynthesis, its regulation and its degradation or breakdown. So start our lecture. कोलेस्ट्रॉल इसमें हम थोड़ा सा इसके इंट्रोडक्शन के बारे में डिस्कस कर लेते हैं कोलेस्ट्रॉल के। Cholesterol, it is a derived lipid. It is a 27 carbon containing compound and it contains hydroxyl group at third carbon and there is double bond in between fifth and sixth carbon. It is also called as animal sterol or animal steroid. अब बात कर लेते हैं इसकी स्ट्रक्चर की। स्ट्रक्चर This is the steroid nucleus and here this is हाइड्रोकार्बन टेल। अब नंबरिंग दे देते हैं हम फर्स्ट कार्बन, सेकंड, थर्ड, फोर्थ, फिफ्थ, सिक्स, सेवन, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, here 18, 19, 20, 21, 22, 23, 24, 25, 26 and 27 carbon. Okay students, this is the structure of cholesterol and it contains cyclopentanoperhydrophenanthrene ring. The important functions of cholesterols are functions. It is very important. It is very important in bile acid synthesis vitamin D synthesis, and steroid hormone synthesis. Students, see, this is a little introduction of cholesterol. Cholesterol, it is a derived lipid, 27 7 carbon containing compound, and it contains hydroxyl group at third carbon, and there is a double bond in between fifth and sixth carbon, and it is also called as animal sterol or animal steroid. This is the structure of the cholesterol. Here is a hydrocarbon tail, and this is the cyclopentanol perhydrophenanthrin ring. This is called a steroid nucleus. Now, the important function of the cholesterols are: it is very important in bile acid synthesis, vitamin D, and steroid hormone synthesis. The chemical formula of cholesterol is C27 H46 and oxygen. Okay students, this is the introduction of cholesterol. अब नेक्स्ट बात कर लेते हैं इसके इम्पोर्टेंट स्टेप्स की जो कि सिंथेसिस में इन्वॉल्व होने हैं. Students, there are 5 important steps in cholesterol biosynthesis. First one is HMG CoA synthesis. Second one is Mevalonate synthesis. Third one is Isopentenyl Pyrophosphate synthesis. Fourth one is Squalene synthesis, and the last one is cholesterol. स्टूडेंट्स, इन स्टेप्स के बारे में, ये इंपॉर्टेंट स्टेप्स हैं जो कि कोलेस्ट्रॉल की बायोसिंथेसिस में इन्वॉल्व होते हैं. अब हम कोलेस्ट्रॉल की सिंथेसिस के मुतालिक डिस्कस करेंगे. There are 5 important steps: hydroxymethyl glutaril coenzyme A synthesis, mevalonate synthesis, isopentenyl pyrophosphate synthesis, squalene synthesis, and last one is cholesterol synthesis. अब हम इसके पाथवे को डिस्कस करते हैं, किस तरह से कोलेस्ट्रॉल की बायोसिंथेसिस होती है, एंड दीज आर द 5 important स्टेप्स. ओके, अब पाथवे को पाथवे देख लेते हैं. स्टूडेंट्स, first of all, two molecules of acetyl CoA, they get condensed and and they form acetoacetyl CoA. But one of the enzyme that involved is thiolase, and here there is a removal of coenzyme A sulfhydryl group. This is the first step. Two molecules of acetyl CoA gets condensed with the help of thiolase enzyme, and there is a release of CoASH, and there is a formation of acetoacetyl CoA. Next one, the next step, by action of HMG CoA synthase enzyme, it converts to HMG CoA hydroxy methyl glutarate CoA, and here there is a involvement of acetyl CoA and release of there is involvement of acetyl CoA and release of coenzyme A. In next step, HMG CoA synthase enzyme, there is a formation of hydroxymethyl glutarate coenzyme A. Acetyl CoA gets incorporated and there is a release of coenzyme A. So student, this is the first step. This is the first step. In next step, by action of HMG CoA reductase enzyme, okay, it converts into mevalonate. It converts into mevalonate. This NADPH is obtained from HMP shunt pathway. इसको हम कार्बोहाइड्रेट के मेटाबॉलिज्म में फर्दर डिटेल से डिस्कस करेंगे एंड दिस इज द सेकंड स्टेप. In next step, kinase enzyme involved, and kinase enzyme, it is very important for the phosphorylation, mean addition of phosphate group to any substance. It converts into this phosphate group attaches to the fifth carbon, 5 phosphomevalonate. Okay students, next one, also kinase enzyme involved. It converts it into 5 prime pyrophosphomevalonate. Okay students, again at fifth carbon the phosphate group gets attached. In next step, again kinase enzyme involved. Kinase. At third carbon the phosphate group attaches and it converts into 3 phospho 5 prime pyrophosphomevalonate. So students, these three steps they are followed by kinase enzyme, and the important function of kinase to add phosphate group in any substance. So in first reaction the phosphate group attaches to the fifth carbon. In next step phosphate group attaches to again fifth carbon, and third step the phosphate group attaches to the third carbon. In next step, no. In next step, the 3-phospho 5' pyrophosphomevalonate it converts into the isopentenyl pyrophosphate and is a 5 carbon containing compound. with the help of decarboxylase enzyme. Okay, students. And the third step is completed. Now, next step. This isopentenyl pyrophosphate converts with the help of isomerase enzyme. It converts to dimethyl allyl pyrophosphate. It is also a fifth carbon containing compound, 5 carbon containing compound. In next step, there is a condensation of isopentenyl pyrophosphate, and it converts into geranyl pyrophosphate. And fifth carbon, and this is a fifth carbon. It is a 10 carbon atom containing compound. In next reaction, there is again condensation of isopentenyl pyrophosphate, and it converts into farnesyl pyrophosphate, and 15 carbon containing compound. In next reaction, there is a condensation of farnesyl pyrophosphate. There is a condensation of farnesyl pyrophosphate. It requires NADPH. There is condensation of farnesyl pyrophosphate, which is a 15 carbon containing compound, and here important enzyme required, that is squalene synthase enzyme, and it converts into squalene, that is 30 carbon atom containing compound. Students, isopentenyl pyrophosphate with the help of isomerase enzyme, it converts to dimethyl pyrophosphate. Then in next reaction there is a condensation of isopentenyl pyrophosphate and converts into the geranyl pyrophosphate. In next reaction there is again condensation of isopentenyl pyrophosphate and it converts into farnesyl pyrophosphate, which is a 15 carbon atom containing compound. In next reaction there is condensation of farnesyl pyrophosphate. It is 15 carbon atom containing compound. With the help of squalene synthase enzyme, it converts into squalene, which is a 30 carbon atom containing compound. In this way, the fourth step completed. Fourth step are completed. Then in next step, with the help of oxidase cyclase enzyme, it requires NADPH and oxygen. It converts into lanosterol, which is a 30 carbon atom containing compound. Okay, students. In next reaction, by action of, now here there is a release of H3OH and two molecule of carbon dioxide, and this reaction also requires NADPH and oxygen, and it converts into ultimately cholesterol, 27 carbon compound. In this way, cholesterol gets synthesized. Remember, I'm repeating again, there are the five important steps. First, HMG CoA synthesis. Second one is Mevalonate synthesis. Third step is the synthesis of Isopentenyl Pyrophosphate. The fourth step, there is synthesis of Squalene. And last, there is synthesis of cholesterol. These are the five steps, and that's how the synthesis of cholesterol takes place. Students, the cholesterol synthesis takes place in liver, adrenal cortex, placenta, reproductive tissues. And the important organelle for the cholesterol biosynthesis is cytosol. And some other steps also occur in the smooth endoplasmic reticulum. Cholesterol biosynthesis takes place in liver, adrenal cortex, placenta, reproductive tissues, and the important organ is cytosol, and some steps occur in the smooth endoplasmic reticulum. Students, this step, it is a very important step. It is a rate limited step, mean slowest step, and it is also a committed step because when mevalonate gets synthesized, then ultimately it leads to synthesis of cholesterol. That's why it is called as committed step. This farnesyl pyrophosphate, it is very important in synthesis of dolicol and in post-translational modification of protein, which helps in the protein targeting. Okay students, अब बात कर लेते हैं regulation of cholesterol. This HMG CoA reductase enzyme is a regulatory enzyme. This enzyme this enzyme is activated by insulin and inhibited by glucagon. And this enzyme favors dephosphorylated condition, and insulin also favors dephosphorylated condition while glucagon favors the phosphorylated condition. This is the first regulation. In another, this is the hormonal regulation. Insulin activates this enzyme, glucagon inhibits this enzyme. In this way, regulation of cholesterol takes place. And other is the feedback regulation. Feedback regulation means the cholesterol, it decreases the synthesis of this enzyme by suppressing the genes that make this enzyme. Okay students, this was all about the biosynthesis of cholesterol and its regulation, hormonal regulation, and feedback regulation. In this way, the regulation of cholesterol takes place. In next my video, I will discuss about the degradation of cholesterol. Thank you.